Hello, I am Professor Sims, and in this video I will discuss the ubiquity of microorganisms, culturing and isolating bacterial colonies. This is the second in the series of 10 lab sessions held as part of my Laboratory for the Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus, course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include learning to perform proper aseptic technique, understanding the meaning and significance of microbes being ubiquitous in nature, learning about the types of media, liquid, broths, solid, agars, slants, deeps, and plates, and becoming competent preparing streak plates and isolating bacterial colonies, being able to observe and describe bacterial colony morphologies, of course, as always, understanding safety and disposal procedures relating to this lab. Microorganisms are ubiquitous throughout the environment, which means that they are found everywhere. One characteristic of microorganisms that contributes to their ubiquity is the fact that they are very diverse. There are three major classes of microorganisms, which are prokaryotes, eukaryotes, and viruses. Prokaryotes are rather simple in their structure. They lack membrane-bound nuclei. They don't have organelles. Usually they are unicellular. They do have a cell membrane and ribosomes and chromosomes. Eukaryotes are much more complex. They have a membrane-bound nucleus. They have organelles. They can be unicellular or multicellular. They have cell membranes, and they have complex cell structures, and they're also usually much larger than prokaryotes. Viruses are really quite different from bacteria in that they have a super simple structure. It's basically just molecular material, DNA or RNA, surrounded by protein and lipid membranes, and they depend on an infected cell or a host. They have to have a host in order to multiply, in order to disperse. Viruses are kind of a point of contention. Many scientists don't agree whether they're even living creatures or not. Another characteristic of microorganisms that contributes to their ubiquity is the fact that they are very old. They've had up to 4 billion years to evolve and adapt and to live in any and all environments on Earth. In fact, the fossil record shows that there have had some form of bacteria on Earth for 3.5 billion years. So for experiment 1, what we're going to do is we're trying to illustrate the ubiquity of microbes in our environment. And our environment is defined as the laboratory itself or the building surrounding the lab. You can choose to sample things outside of the classroom itself, but you should not leave the building. So you choose 2 inanimate objects to test for bacterial contamination. Things like a lab bench or a sink or the bottom of your shoe or the toilet or a door handle. You make note of these choices, designate one, object A, object B. You get a nutrient agar plate and divide it in half, and you swab on one side a swab culture taken from object A, one from object B, and that plate is incubated. And you want to form a hypothesis to see which one would have either more bacterial growth overall, or which one would have more species, or maybe one might have more growth and more species. So you want to have some kind of idea what you think is going to happen and some kind of rationale. And then the instructor is going to we'll do like an open air plate just to see the ubiquity of microorganisms and contamination just from the air, so that's pretty fun. Throughout this course, we will be culturing, that is, growing bacteria. And it may seem kind of counterintuitive to want to grow bacteria because you're used to killing it, but you can't study the bacteria without growing it first. So in order to grow it, you need to know what kind of pH it means, what temperature it grows at, and its oxygen requirements. So here's some guidelines for that. Most bacteria are neutrophiles, meaning that they grow at a neutral pH of about 7. Microorganisms that grow optimally at a pH less than this are called acidophiles. And then at the other end of the spectrum are alkalophiles. These are microbes that grow at a pH between like 8 and 11. Organisms are mesophiles, if they're adapted to moderate temperatures with an optimal growth temperature ranging from room temperature, which is about 20 degrees Celsius, to all the way up to 45 degrees Celsius. Your human pathogens are going to be mesophiles because they typically, they have to live at our body temperature. Our body temperature is about 37 degrees Celsius. So all of your human pathogens are classified as mesophiles. Organisms called psychotrophs, these like cooler environments from about 25 degrees Celsius down to 4 degrees Celsius. Psychrophiles are essentially psychotrophs that can grow all the way down to 0 degrees Celsius. So this is at freezing. They're found in permanently cold environments such as deep ocean water. Thermophiles, on the other end of the spectrum, thermophiles are widely distributed in hot springs, geothermal soils, man-made environments such as compost piles where microbes break down kitchen scraps. Hyperthermophiles, these grow in 80 degrees Celsius up to about 110 degrees Celsius. Noteworthy examples of these are Pyrobolus and Pyrodictum. These are archaea that grow in about 105 degrees Celsius. And these guys are interesting because they survive. Hyperthermophiles can survive many of our regular sterilizing procedures like autoclaving. The growth of bacteria with varying oxygen requirements is illustrated in this figure over here to the right. In the first tube, all of the growth is seen at the top of the tube, and these bacteria are obligate aerobes because they cannot grow without an abundant supply of oxygen. So that's why they're all up here at the top, close to where the oxygen is. In the second tube, the bacteria grow at the bottom. So these are the opposite. These are obligate anaerobes. These are actually killed by oxygen. The third tube shows heavy growth at the top and growth throughout the tube. So this is a typical result for a facultative anaerobe. Facultative anaerobes are organisms that thrive in the presence of oxygen, but they can also grow in its absence by relying on fermentation or anaerobic respiration. The aerotolerant anaerobes in the fourth tube, these are indifferent to oxygen, so they don't use oxygen because they usually have, they usually ferment, but they're not harmed by oxygen either. And then the last tube on the right shows a Goldilocks culture. The oxygen level has to be just right for these bacteria to grow. They can't have too much, they can't have too little. And these are called microaerophiles. They require a minimum level of oxygen at about 1 to 10%, but it has to be lower than the 21% that is found in the atmosphere. So these are fun to grow in the lab.
Microbes are going to need a constant nutrient supply in order to grow in the lab. So if you are running out of nutrients in the media, or if the temperature changes suddenly or anything like that, your microbes can die out. In nature, they would be getting their nutrients and everything from their environment, but in the lab, you have to provide everything artificially. To grow microbes in the lab, we use media, which is also called artificial culture media. The media contains the chemicals and nutrients for microbes that they need to grow. Media usually comes in a liquid form called a broth, or solid forms. Solid forms are going to be agar plates or slants or deeps. Liquid broth is very useful for culturing bacterial specimen quickly. That is, the incubation is usually 24 hours or less, so it's faster than growing on a plate. And that is a pro, right? But a con of that situation is that the bacteria will deplete the nutrients in a broth more quickly than they would in an agar. So it grows fast and it also dies fast. And you can tell that you have growth in a broth culture because the liquid will become cloudy, and the term for that is turbidity. If a broth is turbid, it is cloudy with microbial growth. And remember that broths are great for culturing, but they can't be used for isolation. So this here shows two different broths. This one has a bit more turbidity. It's a bit cloudy. It also shows how there has been a depletion of the nutrients by the microbes that are growing in it. And you can actually see kind of a layer of growth here on the top. This is a new sterile broth that doesn't have any microbes growing in it. It's nice and clear, and you can just kind of see by the color that the nutrients have not been depleted. Next you have agar. Agar comes in different forms, but the word itself, agar, it comes from an agent that's found in seaweed, and it doesn't provide any nutrients, but what it does is it solidifies the media. So you can literally have a broth, you can have a mixture of nutrients and chemicals, and it's in a broth form, and then you add agar to that, and the agar just solidifies it. So anytime you see agar, whether it's an agar plate or an agar slant or agar deep, you know that that is a solid media because it has this solidifying agent called agar in it. Then you usually have plated media that they're typically used for attaining isolation of species. We'll talk about isolation in a bit. They're also used for differential testing and quantifying bacterial densities. Two other types of solid media are known as slants and deeps. Slants are so named because the agar is actually solidified at an angle like this. So it's got a lot of surface area and a lot of exposure to oxygen. Deeps here are solidified straight up and down, and they have very little surface area and very little exposure to oxygen, especially towards the bottom of the tube here. These are inoculated in a stabbing motion, whereas slants you do very similar to a simple streak just on the surface of a slant, like back and forth on the surface like this. Whereas a deep you actually stab it. So you can see here where it's been stabbed and there's bacteria growing where it was stabbed. And the microbes are incubated inside the agar here, whereas on a plate or a slant, the microbes are incubated on the surface of the agar. These are actually placed inside of the agar, and there's much less exposure to oxygen. And because of the way that you inoculate these, you can actually test your microbe for motility. That is, you can observe whether or not the bacteria or the microbes were able to move away from the inoculation point in order to access either more oxygen or more nutrients or whatever. In other words, you can use a deep to see if your specimen have flagella or cilia or some other mechanism for movement. Because microorganisms are ubiquitous throughout the environment, it's very important for students to learn early in this course how to use proper aseptic technique. You do already know about wearing PPE, but in addition to that, aseptic technique involves avoiding contamination of your sample, your workspace, yourself, others. And one very easy thing to do is to make sure that you keep all of your specimen and agar plates and things like that at an arm's length. And you want to keep things covered, keep everything covered as much as possible. You always want to make sure that you're not having things too close to your face, you're not breathing into your plates, don't sneeze or cough while doing your lab procedures, and if you can't help it, then you can sneeze into your arm or wear a face mask. Another aspect of using aseptic technique is using sterile media and sterile equipment. So in a traditional lab setting, your instruments would be sterilized before and after use with either a Bunsen burner or a bactincinerator. So this here is a bactincinerator. This is the base and this is the heating element, and then this would be a metal inoculating loop. And we use these in my on-site labs instead of a Bunsen burner because Bunsen burners have those really big flames, the big open flames, and we use bactincinerators instead of those in order to reduce the risk of fire. You do still need to know how a bactincinerator is used. You would insert a metal loop into the heating element. You want to avoid scraping the sides, so you just push the loop in there towards the back and try not to scrape the sides, and then you hold that loop in the heating element for at least 5 seconds, but you don't want to hold it in there longer than about 10 seconds because you can actually melt the loop. These bactincinerators get up to about 400 degrees Celsius, so you can melt a metal loop in there. So about 5 seconds, no more than 10 seconds. And then once you take it out of the heating element, you want to allow it to cool, and you would not want to cool it by blowing on it. You don't want to set it on the counter because then it wouldn't be sterile anymore. So you just basically the easiest way to cool a sterile loop would be to use sterile media, touch it to sterile agar, and if it sizzles, then it's not ready. If it's still sizzling, it's too hot. But you do want to cool it, because if you cool the loop, then that prevents it from killing the bacteria that you're trying to grow. So if you want to use the loop to transfer bacterial cells from one place to another place in order to grow it, well, you don't want the loop to be so hot that it kills that bacteria. The learning objectives for Experiment 2 are going to include learning to perform the proper aseptic technique, and work with different types of media, being able to observe and describe colony morphologies. And we're going to be using the Saccharomyces cerevisiae yeast for this, and it'll be in lab. You'll be provided one nutrient agar plate, and you will use a sterile, disposable inoculating loop to obtain a sample of yeast from a broth culture that's also provided. Then you will inoculate the nutrient agar plate using the simple streak pattern. Take care not to cut into the agar. You're going to, instead of rolling like you did with the swabs, you're just going to kind of rub the loop very gently across the top of the surface of the agar. And you're going to be sure, as always, to use proper aseptic technique through all of these steps. So you're going to want to keep your plates covered, keep your specimen at arm's length, keep the broth covered as much as possible. All of those things that we discussed earlier. Then after the culture is allowed to incubate, you'll come back and you'll describe the results, including colony morphologies and whether you had contamination and the like. If you have a mixed culture, which a lot of times you will, because in nature bacteria live in mixed cultures, they don't just hang out with their own kind. They're all swimming around together, right? So you have a mixed culture that has more than one species in it. And in order to get a pure culture that has only one species in it, you're going to have to first isolate that specimen. So what you're doing is you're taking a mixed culture or even a pure culture. You can always isolate another pure culture. But the point is that you are going to use a very specific technique in order to obtain isolated colonies. And a colony, you've heard me say the word a few times now, term itself refers to a single cell that has grown and reproduced in such a way that you can observe it, like you can observe this aggregation of cells on a plate with the naked eye. But every cell in that aggregation is genetically identical to each other because they all derived from one single cell that divided and reproduced asexually. The technique itself for doing this, for obtaining individual colonies of pure cultures, is called the streak plate technique, or streaking for isolation. Streaking for isolation consists of using a sterile loop to inoculate an agar plate using a primary streak followed by a second, third, and fourth streak, and sterilizing or switching out the loops between each streak. The inoculum is obtained and then you streak it into the first quadrant right here. So you get your inoculum, streak it, just a little simple streak, but only in one quadrant of the plate. Then you sterilize the loop, or in the case that you're using disposable loops, you would dispose of the first loop and get you a new one. And then you're going to pull just a little bit from the first quadrant into the second quadrant and streak it out. And then if you're using a metal loop, you would sterilize it. If you're using a disposable loop, you would dispose of that loop and get a new one. And then pull a little bit from the second quadrant and streak it out into the third. And then one more time, get you another sterilized loop, and then pull a little bit out from the third into the fourth. So what's happening here is there is a dilution of the cells from one quadrant to the next. And eventually this is hopefully going to lead to you having individual cells that are deposited onto the agar surface. It usually happens in the fourth quadrant, sometimes the third quadrant. But you're starting with tons and tons and tons of cells here, like tens or hundreds of thousands of cells that you're pulling from the broth. And then you're physically diluting that out until hopefully somewhere on the plate you'll have one cell. And then those single cells will propagate until you will have pure isolated colonies on the plate. So this is what they tend to look like in real life. So each one of these little dots that's by themselves is an individual colony, and those individual colonies derived from single cells. So you know that they are 100% pure culture. Experiment 3, you're going to be doing a streak plate in order to isolate colonies of Saccharomyces cerevisiae yeast. And I've also provided a simulation here so that you can go through the steps of streaking for isolation at home and perhaps while you're preparing for the quiz. I do recommend performing the simulation before coming to the lab.
Make sure you go through these safety guidelines. Just a couple things I'm going to make sure I point out here. You will be wearing your gloves and glasses, so make sure you have those. Make sure you have your lab coat. If you have any accidents or spills while you're in lab, obviously you want to make sure that you notify your instructor immediately. Any kind of swabs or anything that needs to be disposed of, make sure that you know where those go. And if you don't know, ask your instructor. Leave anything that needs to be incubated, make sure it's labeled properly with your section and table, and leave those at the end of the bench at the end of class, and your instructor will handle those.
As for observations and interpretations for experiment 1, again, you're looking to see if your hypothesis was supported or rejected. And you're going to be looking at either the abundance of growth or the numbers of species on A versus B in order to see if your hypothesis was supported or rejected. For experiment 2, you're going to look at the density of the growth in the broth, and you're going to look at the density of the growth on the plate. You also are going to do a little bit of literary research because I want you to look up and see what the oxygen requirements are for Saccharomyces and if you should have expected it to grow well in the broth or the plate. And then for the plate culture, the simple streak, you want to have a look at the growth patterns and the morphologies to see if you actually did obtain a pure culture of yeast on your nutrient agar plate, or if you perhaps had some contamination and need to work on improving your aseptic technique. For experiment 3, you're going to be talking about whether or not you have successful isolation of colonies. And you're going to discuss why it was so important to sterilize the loop between quadrants, why it's necessary to cool the loop between quadrants. And again, here are some of the terminologies and things you're looking for when you're describing colony types, colony morphologies. You have things like shape, size, margin, color, elevation. This is an example of what you might see on your Ubiquity plate. And again, it kind of looks like just a whole bunch of dots on a plate, except it's not. You have some things that are semi-transparent. You have some things that are fully opaque. You have smooth margins, you have irregular shapes, you have things that are more of kind of a yellowish color, some more things that are much more of a white color. Here you have an umbonant colony, and you can tell because it's more raised in the middle, flat on the sides. There's another one there. This is a rhizoid. This, in fact, is probably a mold. Okay, so those are some things you look for the differences. You have things that are much larger here, and then things that are very, very tiny. This is a punctiform colony. So at this point, we've really got to get kind of used to describing the morphologies. This is an example of what a Saccharomyces yeast should look like if it's a pure culture. It's got a nice kind of off-white color to it. It's raised. It's pretty large. It's shiny, and shiny is a technical term. Shiny means that it reflects light, whereas dull means that it doesn't reflect light. And this is an example of what your streak plate should look like. It's a simulation, but it shows you what your result is after incubating. So you should have some isolated colonies, either in 3 or 4 at the end, and no contamination. You can tell if it's contaminated if you have growth that doesn't follow the streaking pattern. So maybe something fell in the plate. Or if you have growth, you have colonies that are not all the same. Because this came from a pure culture, it should look like a pure culture at the end. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.